Our hands, used on a daily basis. We probably do not use any other body part as much as we do our hands, starting with activities such as tying our shoelaces to more demanding activities such as playing the piano. Through our highly developed fine motor skills, we are able to perform grasping movements with variable precision and power distribution. In general, the ability to execute such movements is a fundamental characteristic of humans and primates. But how are these processes actually controlled in the brain? And how do we know exactly when to move our hands in order to get a coffee mug? Stefan Schaffelhofer is a neuroscientist at the German Primate Center in Göttingen. His research encompasses neural processes that underlie the various hand movements. Even though our daily grasps appear simple and obvious, their control in the brain is highly complex.
This complexity can be illustrated quite easily by the example of this coffee cup. Initially, visual processes begin and our eyes perceive the image of the coffee cup. This image is propagated to the visual cortex, which is the starting point of subsequent processes that enable us to perceive the shape, form and size of the object. In addition, objects often afford several grasping options, like this coffee cup. We can either grasp it with our index finger and thumb or use all 5 fingers to clasp it.
Within the framework of his PhD thesis, Stefan Schaffelhofer has intensively dealt with the 3 brain areas responsible for the planning and execution of hand movements in primates. The implied regions are known as AIP, F5 and M1. In collaboration with his colleagues Hans-Jörg Scherberger and Andres Agudelo-Toro, Stefan Schaffelhofer discovered during the course of his studies that visual information for objects, especially their 3-dimensional shape and size, are mainly processed in the AIP region. This information is then passed to the area F5, where a motor plan for the hand movements is created. The movement is executed in cooperation with the M1 area.
In order to record the neuronal activity of these brain areas, we implanted so-called micro-electrode arrays. This method enables us to record activity simultaneously from a large number of neurons and to study them subsequently.
Each of the multi-electrode arrays contains 32 hair-thin electrodes with a length of about 1.5 to 7 millimeters. They are implanted directly into the cerebral cortex in the immediate vicinity of individual nerve cells. Active nerve cells communicate with each other through electrical signals that can be measured with the help of electrodes. In order to analyze the grip types of primates, the researchers have trained the rhesus monkeys to repeatedly grasp 50 objects of different shapes and sizes. The objects were mounted on a turntable and presented to the animals in varying order. During the grasping movement, the activity of the neurons in the corresponding brain regions was simultaneously recorded. Based on the so-called delayed grasping task, it was possible to examine the planning phase of the movement separately from the execution.
The object was initially only illuminated, but the monkeys were not allowed to instantly grasp the object, which meant they could initially only plan the movement. In the next step, they were then signaled to grasp, lift and hold the object. In order to determine the correlation between neuronal and movement signals, we recorded both simultaneously, the activity of single neurons with electrodes and the movement of the monkey's hand with the data glove, which was developed also here at the DPZ.
Based on the activity of the nerve cells measured during the planning and execution of the grasping movements, scientists could subsequently draw conclusions on the applied grip types. The predicted grips were compared to the actual hand configurations in the experiment. Since the activity of the measured brain cells is strongly dependent on the applied grip, The neural difference used to calculate the hand movement was used. This step is known as decoding.
The decoding step, meaning the prediction of grip types, is best understood by looking at the activity of a single neuron. Represented in yellow here are single action potentials. We call this neuronal firing. This activity of the cell increases during the movement. Other cells might, for example, encode the planning activity. We extract the firing rate either in the planning period or the execution period and describe the activity of the cell in relation to the grip type. Such observations will be produced for all grip types we want to decode or predict. And as you can see here, this extends from very accurate grip types, precision grips, to power grips. The next step is the actual decoding. Here we record data of grips we do not yet know. Then we compare the firing rates with the data obtained during training in order to calculate the most likely grip type being executed. In this example, a match is the blue category, which means the precision grip is the decoded grip.
Subsequently, the decoded hand configurations were successfully transferred to a robotic hand. This finding is of great importance for the future treatment of paraplegic patients. Therefore, if one knows where and how the brain controls grasping movements, neural interfaces could read these motion signals in the brain during the planning phase, thereby controlling neural prostheses that could be developed in the future.